Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to the past HSC exam question videos. In this video we're going to cover this exam question, which comes from the DNA to protein chapter. While doing a second, I'll read the actual question. Once you have the question, you get about 5 seconds to pause the video. Once you pause the video, attempt the question, and press play when you're ready. The question, there are two parts to this question. The first one is summarize the evidence that led to beetle and tatum's one gene, one protein hypothesis. That's worth 2 marks. And the second part is to explain why the one gene, one protein hypothesis was changed to one gene, one polypeptide hypothesis. And that's worth 2 marks. So when you're ready, press pause and attempt the question. Welcome back. So first question is worth 2 marks, the first one. And obviously we have to talk about the one gene, one protein. So you don't have to talk about the one gene, one polypeptide yet. And it says summarize, so we have to quickly summarize the evidence. And obviously we're going to talk a bit about beetle and tatum's experiment. You wouldn't have to go into detail because it's only worth 2 marks. The main part, you just get the main gist of the experiment, that's what you would have done. And then link that to how that deals with the one gene, one protein hypothesis. So what I wrote is, first I just mentioned the beetle and tatum conducted, actually it's tatum, not tantum, conducted an experiment on bread mold in 1941 to establish a link between genes and proteins. That's just saying that they've conducted an experiment, not going into detail because it's not worth more than 2 marks, so you wouldn't have to go into detail. And then I wrote what they helped establish. They showed that when a bread mold had a mutation induced, that certain enzymes were no longer produced. So we had those mutations induced, we did that for the X-rays. And then what happened is certain enzymes were no longer produced. These enzymes would usually help the bread mold produce in particular amino acids. Remember, because of those enzymes, these amino acids were not produced, especially the specific ones, particular ones, and that meant the actual mold couldn't grow anymore. So that would happen because of the actual UV or X-rays that induce that mutation. And now I'll have the last statement, which says they then managed to link the missing enzyme. So they found what the missing enzyme was. So they managed to link the missing enzyme, which is a protein, to a faulty gene. So the faulty gene was faulty because of the actual X-rays, mutations, and thereby helped establish a link between genes and proteins. So they found the gene, and they knew which protein was missing, and they could make a link between the two. The genes produce proteins. As in this case, it's worth you 2 marks. You've gotten your marks for the first statement. Just quickly, these 2 parts here. It's just really a quick summary of their actual experiment. And the last part was the conclusion of the experiment, or what the actual experiment helped them to do. And now we've gotten you 2 marks with 2. Now for B, explain why the one gene, one protein hypothesis was changed to the one gene, one polypeptide hypothesis. There you have to talk about what genes actually do. So genes produce polypeptides. I might not actually have done that, I should have done that. So you should just say that. And then you should say that some proteins are made out of one polypeptide, but some proteins are made up of more than one as well. So some are made up of one, and then the one gene, one protein hypothesis would hold true. But some proteins are made up of 4 polypeptide chains, where the one gene, one protein hypothesis would not hold true. So what I wrote is, many proteins are made from one polypeptide chain, so the one gene, one protein hypothesis would hold true. And as I mentioned earlier, I should have also written that genes actually produce polypeptides before this statement. This is basically what I had written here, is this statement here. I forgot to write that statement. And then what I wrote is, there are, however, some proteins that are made from more than one polypeptide, such as hemoglobin, so I gave the example as well, hemoglobin, where the one gene, one protein rule would not apply. Right, so in this case, we can't say one gene, one protein for hemoglobin, because hemoglobin is actually made from 4 genes, which help make that hemoglobin. It would be more accurate to say one gene produces one polypeptide. So really, in reality, you probably get, for these 2 statements, you get 1 mark. And then for the one I mentioned earlier, just by saying that genes actually produce polypeptides. So one gene produces one polypeptide chain. that would also get you a statement, except that ties in with the other statements. Where did these questions come from? They came from this experiment. This is the dot point. Students will analyze information from the secondary source to outline the evidence that led to beetle and tatum's one gene, one protein hypothesis, right? That was the first part, more or less straight from the dot point. And the second part was to explain why this was altered to the one gene, one polypeptide hypothesis. Again, that's directly the second part of that question. I hope that was useful.
Thank you for watching.